Hello, it's a real pleasure to be here, and it only took about a year and a half before we found a date that worked. I'd like to talk to you today about genome editing because it's certainly been in the news lately. Many of you may have seen the stories over the last week coming out of China that a researcher there has used genome editing essentially to engineer human embryos and implanted them and brought to term two girls, Lulu and Nana, who purportedly would now have a heightened resistance to infection from HIV. Now, I have a lot of problems with what he did, but I want to tell you first about genome editing very briefly because of its many applications before we get to the really fun stuff about what happened in China at the summit that I'm just back from. So, very fast. Genome editing is basically a more precise way of doing certain kinds of genetic engineering. I'm sure that all of you remember from your high school biology class that genes are made up, chromosomes are made up of genes, and genes are long sequences. And with genome editing, you can actually very precisely add, delete, or inactivate parts of a gene, take out genes, put in new genes. It's really quite extraordinary in its range of potential uses, not just for humans, but also for non-humans like crops or non-human animals. The potential of this was so enormous that it led the US National Academy of Sciences and National Academy of Medicine to put together a study committee, which I co-chaired with Dr. Richard Hines from MIT, and we released a report in 2017, Human Genome Editing, in which for most of the time we said our regulatory systems are adequate, but that sometimes we are a little bit behind the ball, and yet we might still want to go forward with some of the more controversial uses, including so-called germline or heritable editing. So here are some of the key messages from the report that I just want to mention by way of background. You can do genome editing for basic research where you just want to manipulate things in your laboratory dish. And there are a host of regulatory protections against, for example, accidental release or infection of workers or anything like that. Somatic therapy is editing of the cells in your body other than your eggs or sperm or embryos, and it would only affect you during your lifetime. And that is also already regulated through the way in which we regulate gene therapy. We've got the Food and Drug Administration, and we have local institutional review boards that govern that. The really tricky business had to do with the so-called heritable editing, in which you make a change that would then not only affect the person who is eventually born, but also all of that person's descendants. You've now made a multi-generational change. And this was something that was highly controversial from the start. Technically, it wasn't possible for many years, and so the general consensus was that this was wrong. Once it became possible, though, people had to face the question of whether or not they'd give up its benefits in exchange for an absolute prohibition. So, by the way, if anybody really wants this, I'm happy to share the slides because I don't want to kind of go through all of them. So why do we do this? The basic science research is really about understanding real fundamentals of gene functions and regulation, understanding fertility and normal development, and gene and disease connections. And as I said, we have multiple layers of regulation for basic research. The somatic therapy is where I wish we were talking today for the entire 20 minutes, because this is what's really exciting. So somatic editing means taking cells and changing them. You can do it by taking the cell out of the body and change it and put it back in. So there are already people looking at the possibility of doing that for, among other things, HIV resistance, for treating certain kinds of cancers, and very exciting is the possibility of doing this for sickle cell disease. You can also try to do it directly in the body, in which you would do, anybody here a Star Trek fan? Remember Beverly Crusher from The Next Generation? And she was always like poking people with something that went, and immediately they were cured, right? It shot something into their bodies and they're cured. But that's in vivo editing. That's where you would actually shoot something into somebody's body, which would trigger a change in the cells. And we might be able to do something about things like hemophilia, and probably more advanced than anything else right now is being able to increase the muscle strength for boys that have Duchenne's muscular dystrophy. These things are real. We're already in clinical trials for the HIV, for the muscular dystrophy, and for a certain kind of retinal disease, and I think we're about to go in for sickle cell as well. So this is where it's exciting. and where we have a robust system of regulation already in place in the United States, local boards that look at the research subjects protections, a national kind of dialogue center called the Recombinant DNA Advisory Committee, and then the Food and Drug Administration, it's the European Medicines Agency if you're in the European Union, which controls how you begin and end trials and at what point you'd say this is now an approved therapy, okay? This is where the action's at. But we're not, and the only thing that's really become controversial about it is whether you should use it for something that some people would like to call enhancement. You know, I can do something to try to treat your sickle cell disease. We all understand that's therapeutic. And if I were to, for example, take somebody of average height and simply try to make them taller, by the way, that turns out to be hideously difficult. You might call, I'm 5'2", I've been waiting. That would be considered an enhancement because I'm still functional, even if I can't reach the top shelf in my kitchen. I'm functional, so I don't really need this. In between is something like prevention of disease. So every time you've gotten a vaccine, and I hope you all got your flu vaccine this year, you were enhancing your resistance to that particular infection. That's an enhancement. And yet lots of concern about the use of this for enhancements that would somehow increase the inequities in society between the haves and the have-nots. And that's still where a lot of the action's at. But generally, enhancements and even some treatment, some prevention opportunities don't offer benefits that are great enough to offset some of the risks of what is still a very new technology. But we're not going to talk about all the important stuff that I just mentioned. Instead, we're going to talk about this. This is heritable genome editing. This is the thing that excites everybody's attention in public debate. It is now, for the first time in history, a realistic possibility, and it would allow people to have genetically related children without the risk of passing on a genetic disorder that ordinarily their kids would inherit, or at least be at high risk of inheriting. Now, most of the time, there are alternatives. For one thing, you don't have to have a child who's genetically related to you. And many people here probably have adopted children and have experienced the phenomenon of family formed by adoption. Or you could use donor gametes. If one partner has a genetic disorder that is dominant and might express in the children, that person can use donor egg or donor sperm. So half of the parental unit now has a genetic relationship. Or for some people, you could do all of your fertilizations in a laboratory, so-called IVF, and then you can biopsy the embryos and identify which ones have the genetic mutation that's deleterious, and simply not implant those, and instead only implant for gestation the ones that are lacking that. It doesn't guarantee a healthy child. There's a world of other problems people can get, but at least you've now cleared out the one that you can biopsy for. Now, for some people, that's unacceptable because it means you have to discard embryos. For some people, it's unacceptable because it's going to reduce the number of embryos they have available for implantation. But in general, this is going to be a usable technique for many couples who face this problem. Every once in a while, you can imagine a situation where there are no alternatives if you want a genetically related child. Huntington's disease, many of you know a little bit about it, tends to hit in your 40s, 50s, 60s. Woody Guthrie, one of the more famous people who had Huntington's, causes tremors and paralysis over time. Now, with Huntington's, all you need is one copy of the Huntington's, I'll call it variant. I don't want to go into explaining. It's not exactly a mutation so much as it is a number of multiplying certain series of genetic material. But in any event, all you need is one parent and you're going to wind up with the disease. And it's almost 100% certain you'll wind up with the disease. Now, you could get a copy from both parents, have two copies. Now you have no healthy copies. You will still be able to survive for a period of time. So you can imagine somebody who's alive, has two copies of the Huntington's gene, and is married to somebody who might be completely healthy, but every one of their children is going to inherit at least one copy of the Huntington's variant, and that child will have Huntington's. For a family like that, there is no alternative if you want a genetically related child except to go in and actually edit that embryo or edit the gametes in order to change this to a normal variant, what would be called wild type. So it's for things like that that one could imagine a very small number of situations in which genome editing makes a lot of sense. But it has also raised a lot of concerns. First, because you have genetic changes that are inherited by the next generation, you actually have multiplied the degrees of uncertainty about risk as well as benefit. You also have concerns, again, about a kind of eugenic mentality and a creation of haves and have-nots. You also have reasons why people have thought that this was a problem because of the lack of consent, because future generations never agreed to be edited and changed, right? And then I think probably cutting closest to home, because some of the objections are more instinctual, is the idea that somehow this is playing God. It's taking too much control over our human reproduction. And for many people, there's an instinct that somehow this should not be within our control. And often these tend to reflect different religious traditions. I remember years ago when I was on President Clinton's Bioethics Advisory Commission, we were talking then about cloning, and we had a battery of people from different religious traditions. And coming from the Christian and like Protestant and Catholic traditions, we heard a lot about finding grace through the acceptance of things. When we got to the rabbi and the imam, we began hearing very different things. Rabbi Tendler, I love Rabbi Tendler, said, from our tradition, man is a guest in God's house, and what does a good guest do? Doesn't wait for the host to serve him. Guest gets up. He literally said, guest gets up, goes in the kitchen, gets a cup of coffee, gets the orange juice, helps himself. And he said it just like that. He said, but they don't move the couch, right? You can look at the transcript. We had a conversation at the table about whether cloning was moving the couch or getting coffee, right? Oh, that was my New York accent coming out, wasn't it? And finally, the idea that somehow it's a step toward designer babies, in which we treat our children as products to be designed, and that our love will no longer be unconditional, but we will reject the defective product. Now, I gotta say, having worked through this with our committee at the National Academy of Sciences, I found that if you look back over the last 50 years, these are exactly the same kinds of concerns that have been raised about in vitro, first about amniocentesis in the 1970s, then about in vitro fertilization and sperm donation with, kind of screened sperm donors in the 1980s, pre-implantation genetic diagnosis in the 90s. Every new technology that gives us control tends to raise these same concerns. And with very few exceptions, I am relieved to tell you that these things have not come to pass. We have not created, kind of biological caste system. We have not, in fact, created designer babies. Parents still love their children the same way they always did. So it gives me some comfort that with even this new level of control, we don't necessarily see these kinds of problems coming to pass. Now, there are lots of regulations that apply, and I don't want to go into all of them because of the time, but I want to simply say that we do have controls over this, and believe it or not, in the United States, although it doesn't sound like it should, it's under the Food and Drug Administration, because it would involve the transplantation of an edited embryo, which is the transplantation of a manipulated human cell or tissue, which the FDA regulates. And right now, it is not possible to do this in the United States, no matter how hard you try, because the Congress has prohibited the FDA from spending any of its resources, including staff time, to review an application to try to use this in a clinical trial. So right now in the US, this is functionally illegal. That might change, we'll see. But even if it were legal, our committee had concluded that we really need very strict criteria for this because of all the uncertainties about it. For one thing, we need tremendous amount of research now on animals in order to be sure we understand when you make a change, whether it's going to change every cell in the embryo or only some of them. Only some of them means you get what's called a mosaic, and it can have implications we don't even understand yet. Or so-called off-target effects. I want to change this particular gene, but it might have an effect on a neighboring gene or something even further distant, and we need to understand those effects. And caution, because sometimes what is an unfortunate trait also has a bit of a silver lining. So for example, with sickle cell, 2 copies of the sickle cell trait and you have the disease, which is incredibly painful and life-shortening. 1 copy of it, however, you don't have the disease, but you may have some enhanced resistance to malaria. There's some suggestion that cystic fibrosis, which is one of the most common genetic disorders in the population of people originating in Northern Europe, might also have offered some protection against certain diseases that were ravaging Europe in the Middle Ages. And so you need to be careful about making changes because you might actually inadvertently swap one problem for another. Now, this is, of course, what was in the news last week. This is the story of a Chinese researcher, He Jiankui. Everybody's pronouncing his name differently, so I'm still not sure. And I actually met with him. But he goes by JK, which is a lot easier. I mean, really, no, it's Dr. He, Dr. He, Dr. He. And, and none of us from the West could deal with like the tones in Chinese, which, really distinguishes these things. So we all sounded like idiots. We were in Hong Kong last week for what was going to be the second International Summit on Genome Editing, a serious scientific meeting about the new possibilities, a great deal of emphasis on somatic editing and where we are in clinical trials, what the regulatory system is like, how can we foster multinational trials if the rules are slightly different from one country to another. This was a serious meeting. And about 36 hours before the meeting began, we got the news that a researcher in China had decided he was going to edit some embryos and bring them to term. And clearly, his timing of all this was very deliberate about trying to get maximum exposure. This is a JK at the summit. It's really hard to see. I'm sorry. The bottom is a photograph I took on the. If you look at right by the wall there, it's just a sea of journalists. So we were swamped by the press. What he did, what he claims he did, because it still hasn't been validated, is the following. He said, HIV and AIDS are a scourge in China, and I want to do something about it. So he recruited families in which one parent was HIV positive, and in particular where it was the father who was positive, and said to these couples, why don't we do IVF? We're going to edit the embryos in order to delete a particular section of the so-called CCR5 gene. Why? Because that particular deletion, so-called the delta 32 deletion, is present in a fair number of people in Northern Europe, and they seem to have a resistance to HIV. And what we'll do is we'll edit them, we'll bring them to term, and now your children won't be at risk. Now, there are a few problems with this. First, the animal data is not ready yet to understand how to make sure that your changes are on target and how to avoid off-target effects, or how to evaluate the off-target effects that you do find. This is what we might call regulatory science. It's the boring, non-sexy, don't get tenured for it kind of science that you need. That's one of the real problems, that you need for the medical reviewers at places like the FDA to have standards that they can look to and say, is this safe enough? Do we understand enough? And we don't have anything close to that amount of so-called preclinical research yet. So that was one of the first problems. Another one of the problems with what he did is that one of the most important criteria that we had mentioned in our report, going back just for a moment, was the idea that there's an absence of reasonable alternatives, and that we're dealing with something that is a serious risk, a serious disease, and has a compelling need. Well, here's the dilemma. It's incredibly unusual for children to contract HIV when the father is the one who's positive if you take the simple step of what's called sperm washing, which he was doing in his so-called protocol anyway. So these kids were going to wind up HIV negative no matter what. And you really want to worry about HIV transmission from parent to child. What you worry about is the HIV positive mother, where the fetus is developing within her system and the exposure is much greater. Even there are reasonable alternatives. We have drug regimens now that can reduce that risk profoundly. So you don't need to do genome editing in order to reduce this risk. But he did it anyway. Now the next problem is that he didn't actually edit it to become what he claimed it was going to become. You may remember, with all the kind of weird numbers and letters I've been mentioning, that the variant in Europe that gives you HIV resistance is called the delta 32 deletion of CCR5. Well, his edits seem to have had a somewhat random quality to them. He did edit in the CCR5 region, but he didn't get the delta 32 deletions. He got deletions of 15 base pairs, 23 base pairs, none of which have been documented to offer the same level of resistance as this particular deletion that we see in Europe. The next, because genes don't operate in isolation, what is a useful variant in Europe might be problematic when it is in a different region because the genes interact. So it's possible that this particular deletion, even if you'd achieved it, wouldn't give you the same resistance because it might depend upon interaction with something else that has a different frequency in Asia. Even more problematic is, as I said, genes can have both positive and negative effects. So those with the CCR5 variant are actually more vulnerable now, even though they're resistant to HIV, they're more vulnerable now to West Nile virus and possibly influenza. Now, West Nile virus is not common in China, but influenza is a major problem because you might notice how many of our flu seasons have a name of a flu that seems to have originated somewhere in Asia, often through contact with animals. So one of the last things you'd want to do is increase the risk of a severe reaction to infection with influenza. Next, when there were English translations now of the consent documents. We don't know what these people were actually told when they agreed to do this, but when you look at the English translation of the document, it seems to summarize it, it suggests that they were told that this was an AIDS vaccine trial. It wasn't. That they were told it would likely have benefit, but there was no benefit to be had. The kids were going to be negative no matter what. And very interestingly, IVF is very expensive in China, but if you agreed to participate, you get your IVF for free, which is a very strong incentive for people who have no interest in the research or no need for this particular kind of protection, but do want to get IVF services. And he failed to abide by a whole variety of rules. Now, my head is exploding trying to understand Chinese law. In the United States, the speed limit is posted at, let's say, 60 or 65, but the actual speed limit is how fast can you go before the cops will stop you, right? Law in action, according to my law faculty. Well, everything in China seems to have that flavor in which there's tremendous discretion about how the laws and guidelines are actually utilized. So it appears that there is actual legislation or executive decree, in China that would make his work illegal, but whether it's actually used is unclear. He claims he got permission from local bodies, like the hospitals that he was working at. The story only gets stranger because they now say they never gave permission, that the documents are forged. It goes on and on and on. So this has now become the case that's going to spur the next big debate about the legality of heritable germline editing. In other words, are we going to make policies in which we make this permanently illegal because we're reacting to bad cases like this, or are we going to try to develop a responsible translational pathway that sets out the kind of preclinical research you need to understand the risks and benefits, and then limits the uses to compelling circumstances without good alternatives under strict scrutiny from local bodies, national bodies, and then, most importantly, long-term follow-up, so that even if a child appears healthy at birth, you want to make sure that you haven't missed something that won't appear until later in life. That's a tall order. And I don't think that this kind of editing is going to happen very often, even in places where it's legal. But the biggest problem now is simply the fear that this is going to be one of many examples of things where the first tendency for people who are worried is to say we should have a law that makes it illegal and shuts down a whole field of scientific research. For me, even more problematic would be if, incidentally, the drafting of that legislation were to roll in other things that are not nearly as problematic and are very promising. Cloning, when it happened in the 1990s, led to state legislatures writing bans on cloning that not only banned reproductive cloning, but as an incidental matter, banned laboratory research that had nothing to do with children. I used to give a talk from stem cells to jail cells in honor of colleagues in Iowa who were hit up with something like that. So it's possible to have just bad drafting that has all kinds of effects that go beyond what you want. So I wish that I could say that you are now very well educated about somatic genome editing and all of its great potential. But instead, I'm happy to take your questions about the Chinese babies and the edits that were done on them. Thank you.
Great job. I'm Willie Larkin, and I wanted to ask you, could you speak very briefly about the ethical issues of the Johns Hopkins study on cells with Henrietta Lacks? And if her family should get resources or residual from all of the millions of dollars that those cells brought?
So for those of you who are unfamiliar with the story of Henrietta Lacks, she was a patient in the 40s and 50s. And it turned out that some of the cells in her body had unique characteristics that were terribly useful for research. And some of those cells were retained and ultimately became widely used for all sorts of purposes, academic and industrial, nonprofit and for-profit. And her family did not necessarily know anything about this or get any of the financial benefits for it. I think that there's a very big ethical dilemma about taking material from people without getting their permission or having them at least understand that this may be something we're trying to encourage as a new form of civic duty. And I certainly think that that is a problem in that story. When that happened, I should tell you that our notions about consent for people who are subjects of research were very different. And it was not at all the norm to get consent. That is something that really started after World War II in reaction to the events in Germany and in Japan with so-called medical experimentation, which was simply a form of torture, and led to the Nuremberg Code. So I think the consent issue is very clear. I think the financial one is actually a little bit more complicated because I start from the premise that people shouldn't have to have a reason to get health care and healthcare financing. They should just have health care and healthcare financing. So the unfairness already starts with the fact that you'd even need money from your cells in order to be able to afford to go to a doctor. The second, though, is that we don't have a good theory about how we relate to our own bodies. You know, my tissues, if those tissues are being used by Rob over here, and he has manipulated them and he's added value to them through his own intellectual endeavors, how much of that is me and how much of that is him is really not determined. And I would like to see this be consistently determined because we have lots of other situations in which people should or should not have control over what happens to the tissues and should or should not have a share in the value of what happens to them. I think that because this story has such a resonance, these cells turned out to be so crucially important and are used so very widely, that there is, maybe as an ethical matter, if not as a legal matter, an obligation to give something back to her family because we can no longer give something back to her. Legally, I'm not nearly as clear about it, but as a matter of comedy and respect, I agree that she should be compensated, or the family should be.
Yeah, thank you. A great talk. I had a number of points, but I've been told I don't have much time here. But I think one thing that's really important here is you've emphasized that most of the research in this area is in somatic mutations in those changes. But now what he's created is germline mutations in these two girls. So they'll carry these variants not only in all of their tissues and all their organs, but also in their ovaries. Yes. And so they will biologically pass this down to their offspring, or it will create a dilemma for them as to whether or not they'll have offspring.
That's exactly the point of heritable genome editing. It's multi-generational, yes.
Absolutely. The other issue about this is it's almost Frankensteinish, if you will, and it's patina, in that he could not get this published, obviously, in a peer-reviewed manner.
He didn't try.
And he just did this on YouTube.
He didn't try. I had dinner with him the night before the summit began, along with a few scientists, as we were trying to find out what exactly had he done. And he did publication by press because he thought that his work was so important it couldn't wait for peer review. Yes, the scientists in the community had exactly that reaction too. It's like, are you kidding me? But that was his excuse for it. And he claimed that it was entirely transparent because, of course, it was going out to the press. But in fact, transparency in this context also includes revealing your methods and your data to people who are able to critique it. It wasn't until at the summit when we allowed him to speak so people could hear something that he put up his slides. We're all taking photographs of the slides, showed the data, and that's how some Australian and actually a geneticist in Utah figured out that he hadn't actually done the deletions he claimed that he had done, but had done something else. And it was only because we were able to finally see some of the data that it was possible for the scientific minds there to figure out where he had gone, well, one of the ways in which he had gone wrong.
One last question.
Hi, Ken Yoska is my name. There's other ways to have disturbances of the gene pool, and a lot of them are heritable and need regulation. The other day I saw in the newspaper there was some fellow in Europe that took up a hobby of being a sperm donor, and he had several hundred children, like maybe 500. And I thought that was a little over the top.
Oh, more than a little, yes.
How do you regulate this stuff? And what is his responsibility? What is society's responsibility? And how should this be handled?
Oh, wow. Okay. And we've got about 45 seconds left. Yes, the gene pool is constantly changing through natural mutation. And in fact, what happened to these girls, their descendants will ultimately probably change too. How do we regulate? It requires a lot of oversight. And who the hell missed this guy with 500 kids? We have rules in the United States that actually will limit the number of donations that sperm donors can make. Some of them are informal. They're administered by societies for reproductive medicine. But we have had, in fact, a couple of missteps. There was a doctor in Virginia a number of years ago, Cecil Jacobson, who was providing anonymous donor sperm to women who needed donors, not realizing that he was using his own each time, and wound up with 77 children. And they only figured it out when women were going to the doctor and looking around the waiting room and thinking how remarkably similar, seriously, how remarkably similar their children looked, right? He was prosecuted. He was the U.S. Attorney for the Eastern District of Virginia, was trying to figure out what he did that was a crime, since technically it was an anonymous donor, and they hadn't said you may not use this. And he figured it out that he had phone calls and letters going from Virginia to Maryland, D.C., and therefore he was convicted for mail fraud. M-A-I-L, not M-L-E, M-A-L-E. I think at that I should probably stop. Thank you.
Thank you, Professor Charo. On the way out, don't forget to look at the gift tree and possibly pick up a tag. We are adjourned.